Good afternoon, learners. Welcome you all again to this one of my next sessions. If you remember, in my last sessions, I actually dealt with BBYET 143, that is Economic Botany and Plant Biotechnology, and already I had quite a number of sessions on several units of this particular course. Now today I will take up another topic, that is construction of DNA libraries and genetic engineering methods, and this is also a part of this course, that is Economic Botany and Plant Biotechnology, and you will find most of these discussions in your unit, that is recombinant DNA technology. I already dealt quite a number of topics on recombinant DNA technology in my earlier sessions, but I thought that some of the topics might need to be elaborated in order to make you better understand, and therefore today I have taken this topic which belongs to this unit, that is recombinant DNA technology. Now you will see from today's title that actually there are two components. One is construction of DNA library. So library is one component, and the second component is genetic engineering methods. So there are two topics which need to be discussed, and I will try to divide the time accordingly and discuss or make you understand these two particular topics. Now to start with, what is meant by a library or a gene library? Now, all of us know what is meant by a library, because you know that when we go to it can be a book library, it can be a video library. So library is a place which consists of a collection of certain things. If you go to a book library, then you find a collection of books. If you go to a video library, you will find a collection of different videos or CDs maybe. Then what is gene library? So the concept is that a library is such a concept where you actually have a collection of all genes. Let's take the example of any organism, human or any animal or plant, like let's say Arabidopsis or rice. If somebody asks you the question that what are the genes which are present in rice? I mean, it's very difficult to answer this question because there are so many genes. How many genes are there in human, and what are those genes which are present in a human system? The answer is it's a huge number of genes. So you should have some component, or you should have some way out by which you can actually mention or you can actually describe the gene pool which is present in an organism. And that is what a library means. It's a collection of genes. Now, obviously, we need to prepare this library. That is, experimentally, you need to construct a library. It's not ready like you construct a book library. You have to construct it. You have to go for the collection of books. You have to keep them in proper order. You have to catalog them. So similarly, here also you have to create, and that is through proper experimentation. You have to create a library. Now, when we are talking of a gene library, then there are actually two components. One is it can be a genomic library or it can be a cDNA library. What is the difference between the two? That is, when you are talking of a genome library or genomic library, you are dealing with the entire genomic component. That means in a genome, there are both exons as well as introns. Exons which are actually expressed and introns which are actually spliced out during RNA splicing. But when you talk of a cDNA library, you don't have the intronic sequences because you get the cDNA from the RNA. Because when we prepared the cDNA, we actually prepare it from RNA using one particular enzyme, which perhaps all of you know, that is reverse transcriptase, for which Nobel Prize was awarded for the discovery of reverse transcriptase. So you construct the cDNA library, and from the cDNA actually you try to clone the cDNA. Like you clone the genomic sequences to construct a genomic library. Similarly, you clone the cDNA sequences to get a cDNA library. But in any case, they are actually different forms of gene library. So my first slide or my first PowerPoint presentation will show that what is meant by a gene library. It's a random collection of cloned fragments in a suitable vector that ideally includes all the genetic information of that species. And this is also called the shotgun collection. There are two ways by which gene libraries are made, referred to as complementary cDNA and genomic DNA libraries. Genomic libraries and clones are necessary in addition to cDNA clones because cDNA are generated from processed mRNA which lack introns and other sequences surrounding the gene. As I said that the cDNA, they only represent the exonic sequences, that is the expressed sequences, whereas the genomic sequences, they represent both, that is the exon as well as the introns. So there is a basic difference between the two. The genome is remarkably complex and a particular fragment comprises only a small fraction of the whole genome. Therefore, construction of a useful and representative recombinant genomic library is dependent on the generation of a large population of clones. So I hope you got the idea what is meant by library. So library, now if I define it in a better way in terms of molecular biology, then a library actually represents a series of clones. So what you have to do is, let's say if I ask you to prepare or create or construct a genomic library from rice or Arabidopsis. Obviously, you have to understand that you have to start from the genomic DNA. So you have to first obtain the genomic DNA or chromosomal DNA of that particular organism, whether rice or Arabidopsis, fragment it, that is, you digest the DNA, okay, partial digestion of the DNA using certain restriction enzymes, restriction endonucleases, and then you have to clone it. Normally, for cloning, we use vectors like plasmids, cosmids, bacterial artificial chromosomes, yeast artificial chromosomes, and so on. There are so many vectors. Bacteriophage can even be used as a vector. So there are so many vectors, and each of these vectors, they have their own capacity. Like the plasmids, they can accumulate smaller fragments. Cosmids, a bit larger. If you go for bacterial artificial chromosomes or yeast artificial chromosome vectors, they can accommodate even larger fragments. So when you are dealing with a genomic library and you are isolating the genomic DNA, digesting it with restriction enzymes, what is your expectation? What do you expect to see? You expect to see a heterogeneous group of fragments of different sizes, some of which may be large size fragment, some of which may be small size fragment. But mind that all these fragments need to be cloned. I already discussed about the cloning procedures in my earlier sessions. What are the steps for cloning? I have already discussed. If you have followed that, then now what you are doing is, the difference is that here you are not cloning one particular fragment, but you are cloning a series of heterogeneous fragments. So it's a more kind of random process. And not only you are not using the plasmid vector, because the capacity of a plasmid vector is much less, because some of the fragments can be huge, large size fragments, and for that you need to use some vectors with high capacity. So they can be bacterial artificial chromosomes or yeast artificial chromosomes or maybe cosmids. So such kind of vectors need to be used to accommodate the heterogeneous fragments generated as a result of restriction digestion of genomic DNA. And then you have to select those clones, and then you have to identify or sequence those fragments and see what it is. There are several ways by which you can actually understand what type of gene it represents, that fragment represents. I'll come to that. So if somebody asks you now, which is very important to know, that what are the steps for generating a genomic library? So I will now discuss that point by point, or the steps. The first step is you need to know how to isolate the genomic DNA. So let's say rice, wheat, or any plant which is given to you. So you have to first learn how to isolate the genomic DNA from that plant sample. That is, of course, a part of the practical course, but to be very precise, there is one particular method which is known as the CTAB method, cetyl trimethyl ammonium bromide method, which is a kind of detergent, and you use that detergent, okay, in a suitable buffer and use it for homogenizing the plant tissues, and then through series of subsequent steps like centrifugation, phenol chloroform extraction, precipitation through alcohol, isopropanol or ethanol in presence of a salt like sodium acetate, ultimately you precipitate the DNA, and then in that way you isolate the DNA. So the principle of isolation is based on the CTAB method. Of course, there are other methods also for DNA isolation, but that is most commonly used irrespective of plant specimens. Rather, it works for all the plant specimens. So that is the first step. You need to know and you need to learn how to isolate a good quality, non-degraded genomic DNA with intact bands. Once you learn how to digest the genomic DNA, the next step will be you have to digest the DNA. That is, you have to use a restriction enzyme to fragment the DNA, because otherwise how will you clone? And moreover, you want to have a representation of the different genes. But mind that in this case it's a high molecular weight DNA. So you need to digest it, and in most cases you go for partial digestion using enzymes like SAO-A3 to obtain fragments with desired range of 9 to 25 kilobase pair, with about 30% of the genomic DNA remain undigested. Mind that this is a very important term, partial digestion. You do not go for complete digestion. 30% of the DNA you remain it as undigested DNA. So you get a series of fragments. Now what will be your next task? Next task will be to clone these fragments. And obviously this will be a very random process, because each clone will represent a different size fragment. Unlike a normal cloning, like cloning one particular gene in a particular vector, there the gene is fixed, but here it is not fixed. Here it's a series of fragments, assuming that each fragment represents something new. So you are cloning it, and then you choose the vectors. What are the vectors? All of us know that plasmid is a very common vector, but mind that in this case plasmid may not be suitable for you, because the accommodation power or the potential of plasmid vector to incorporate very large size fragment is very poor. So in this case, we have to choose some high capacity vectors. Now what are those high capacity vectors? Bacteriophage lambda and cosmids. They can accommodate large DNA molecules. Bacteriophage lambda has high cloning and packaging efficiency and is easy to handle and store as compared to plasmid vector. So the capacity is high, you can use cosmids. Cosmids can accept approximately 45 kilobase of foreign DNA, almost twice as much as bacteriophage vectors. But there may be certain genes which are even larger in size, and even bacteriophage or cosmids may not be suitable. So as I said, in these cases you have to actually go for super high capacity vectors, and those are actually covered by the BAC, that is bacterial artificial chromosomes and yeast artificial chromosomes. So you have to make a proper choice of the vector if you really want to make this work successful. Now once cloning is done, next step is, for cloning what you need to ligate. Ligation, that is joining. The fragment, whenever you want to clone any fragment or insert any fragment in a vector, it has to be joined, and for that you need to use one enzyme, that is ligation. That is, the enzyme is called DNA ligase, the process is called ligation. And so the next step is ligation of the fragment to a vector. But again I repeat, this is not a fixed size fragment because the fragments are of different sizes. The non-essential region, like if you are choosing lambda phage as vector, what you can do is the non-essential region of the phage genome is removed with the same restriction enzyme used for digestion of DNA. The vector arms are purified. These vector arms and the genomic fragments are annealed together. So you use the same restriction enzyme to remove the unwanted sequences from the phage genome. Okay, and then you choose the ligase enzyme, go for the ligation of the fragments, heterogeneous fragments, into the bacteriophage vector. And ligation conditions, those have to be maintained. Like in the laboratory for routine cloning, what do? Normally we go for ligation, like we can set the ligation reaction in a bit cold condition. Normally we set it at 20 degrees centigrade or 16 degrees centigrade. You can set the ligation reactions overnight also. Or sometimes some manual, they actually represent ligation reactions to be successful or to occur within 4 to 5 hours. Or you can extend it to overnight also, depending on the temperature. So ligation is done. Next step is packaging. The resulting bacteriophage lambda are packaged by using packaging extracts which contain head and tail proteins of the phage and endonucleolytic cleavage enzymes. Packaged particles can then be added directly to a culture of bacteria. The phage particles are used to infect E. coli for initial selection of phage particles containing recombinant DNA only. So once the phage particles are made, then you infect E. coli cells with the phage particles and then go for the selection, because we can select the E. coli. You cannot go for the selection of the phage. You have to select actually the E. coli cell. How do we do the selection? If you remember my earlier discussion, you can select it through antibiotic screening. So that is the principle here. So you need to infect the E. coli cell, okay, and for the initial selection of phage particles containing the recombinant DNA. So packaging is done. Now you need to identify the clone genes. Now finally, when you infect the E. coli cell, get the colonies, final question is, okay, then what is the clone? What does that clone represent? How do you do that? So you have to now analyze the DNA to yield information about gene structure and function. Now most of these procedures, that is identification or screening you can say, they are based on a technique of hybridization probing. Now what is meant by a probe? When we talk about southern blotting, northern blotting, we have the concept of probe. What is probe? Probe is a piece of DNA which is labeled. Okay. Supposing if anyone asks you that whether, like let's say I ask you a question, like you tell me whether rice genome has catalase gene, because catalase is an antioxidative enzyme and ideally it is present in all the systems. But if somebody asks you, okay, can you show me experimentally whether the catalase gene is present in the rice genome? Yes, that is possible. How is it possible? You can use this hybridization principle. What will you do? You just take the catalase gene, you take the DNA sequence of the catalase gene and you radiolabel it. That is using alpha 32P. And then what you do is next you isolate the rice genomic DNA and now you try to hybridize, you try to bind that probe, that is the radiolabeled catalase gene, that sequence. You try to hybridize it or you try to bind it with the rice genome. So if rice really has that catalase gene inside, it will bind to the catalase, it will bind to the probe because it will find its homology. There is a sequence homology, so it will bind based on the homology, it will bind. And therefore, when you develop the X-ray film, you will find the band. This is actually the principle of southern blotting, where using this experiment we can determine whether a gene is present in a genome or not. The same principle you are applying here in order to screen the clones. Let's say you want to find out whether the catalase gene is there in this library. The same thing I can ask here. So what you do is, in a similar fashion, you take that catalase gene as a probe and you try to use this probe in order to screen the library. So if it binds to the isolated DNA from any of the clones, that means the gene is there. So this principle is known as the hybridization probing. So a probe is a molecule, DNA or RNA segment, that recognizes the complementary sequence in DNA or RNA molecule by molecular hybridization, and thus allows identification and isolation of the specific DNA sequences from an organism. So it's based on hybridization. Hybridization with what? Hybridization of the probe. So that is all about the preparation of genomic library. That is, what are the steps for the preparation of genomic library. Next we come to the cDNA library. What is the difference between genomic DNA and cDNA? I already told you. The cDNA will represent only the expressed sequences. How do you prepare cDNA? That is from the RNA. So what you need to do here is, you need to isolate the RNA, not unlike the genomic library where you are isolating the DNA. You are isolating the RNA. And RNA isolation is a very, very, tricky process. RNA is very sensitive. So you have to be very, very careful when you are isolating RNA from any specimen, particularly from plant system. It is more difficult because plants have different reserve material, secondary metabolites, which actually interfere with this isolation. Often the RNA gets degraded. It is so sensitive. But that is a different topic of discussion. Okay, the practical part where you need to learn how to properly isolate an RNA. I'm not going into that details, but the first step is you need to isolate the RNA. And from that RNA, people can do it, people synthesize cDNA from RNA in two ways. Either you can use total RNA, or you can go for purification of total RNA into messenger RNA. How do you exclusively prepare messenger RNA from total RNA? Because you know that the messenger RNA has poly A tail, AAAA, and so on. So you can use this as a principle to separate out messenger RNA from total RNA using oligo-DT cellulose column. So if you use an oligo-DT cellulose column, then only the messenger RNA out of the other RNAs like ribosomal RNAs or tRNAs, only the messenger RNA will get trapped, and then you try to elute the messenger RNA or try to release the messenger RNA from the oligo-DT cellulose column. So it's based on a chromatography principle. Once the messenger RNA is being synthesized, next what you do is you go for cDNA synthesis using one particular enzyme called the reverse transcriptase, and this is popularly called RT reaction, reverse transcriptase reaction. So isolation or extraction of mRNA is the first step. So obviously you need to select a proper tissue from where you are preparing the RNA, right? From where you are actually trying to create the cDNA library. That is very important. Second step is synthesis of the first strand of cDNA. So how do you prepare it? By using reverse transcriptase. And I already talked about the oligo-DT cellulose column, as you can see here, because only the messenger RNA will bind to oligo-DT because they have the poly A tail at the 3' end. So this is actually the diagram for the preparation of the first strand cDNA. So messenger RNA use the reverse transcriptase and you get the cDNA, but here what you get is a cDNA mRNA hybrid, from where you need to synthesize the second strand cDNA. This is the first strand cDNA that you have made. It occurs in a stepwise reaction. First, you synthesize the first strand cDNA, giving rise to cDNA mRNA hybrid, and then what you need to do is you need to synthesize the second strand cDNA. So the synthesis of second strand cDNA, there are two methods. One is self-priming, which is not that commonly used. The mRNA hybrid so obtained is denatured so that a second strand can be synthesized on the single strand of cDNA by the Klenow fragment of DNA polymerase I, which is self-priming. Due to unknown reason, the single-stranded DNA has a transitory hairpin structure at the 3' end, which can be used to prime the synthesis of the second strand of cDNA. If you look at the picture, probably you can understand better. See, on the this 3' end, you have a hairpin loop-like structure, and this hairpin loop is acting as a primer to synthesize or to go for the synthesis of the second strand cDNA. And once the second strand cDNA is made, then finally what happens is there is a digestion using the single strand specific S1 nuclease, the hairpin loop and any single stranded overhang which is present at the other end are digested. So this was there. This is finally digested using S1 specific nucleus. So final product is a double stranded blunt ended DNA molecules. So this is how you get a second strand of cDNA. So this method is known as the self-priming method. But as I said, that self-priming method is not that much common. The more common method is the replacement synthesis. what happens here? The cDNA mRNA hybrid is used as a template for a nick translation reaction. RNase H produces nicks and gaps in the mRNA strand of the hybrid, creating a series of RNA primers that are used by E. coli DNA polymerase I during the synthesis of the second strand of cDNA. Again, if you see the picture, you can find it out. See the picture carefully, that here you are adding the RNase H, and this is the E. coli DNA polymerase I. So you are adding all the DNTPs, and the nicks are being produced, as you can see here. So the remnants of mRNA serve as primers for the synthesis of the second strand of cDNA. And then using the ligase, there is the joining, and you finally get a double-stranded cDNA. So the reaction has three advantages. It is very efficient. It can be carried out directly using the products of the first strand reaction, and it eliminates the need to use nuclease S1 to cleave the single-stranded hairpin loop in the double-stranded cDNA. Finally, again you have to go for cloning. So once you have completed the synthesis of second strand cDNA, that is double-stranded cDNA, now you can clone it very easily. So cloning, what you do is, one difference between genomic library and cDNA library is that in case of cDNA library, the fragments are comparatively smaller in size as compared to genomic library, genomic fragments which are huge. So in this case, probably you can also use plasmid vectors. It's not mandatory that you have to go for YAC or BAC. You can go for small capacity vectors also. You can go for plasmids, you can go for cosmids also. Or maybe lambda, bacteriophage lambda. So the recombinant plasmids are used to transform bacteria, usually the E. coli K12 strain. So you can use plasmid, as I said. Then you prepare the competent cells, E. coli competent cells. You can use calcium chloride, chilled calcium chloride to prepare competent E. coli cells. And these competent cells can take up recombinant plasmid. I hope all of you know what is meant by recombinant plasmid. This has been discussed already. And then you go for the selection. That is, you select the transformed bacteria on antibiotic medium, because you are using plasmids in this case, so no problem. The plasmid will have a certain antibiotic resistance gene, and you can add that antibiotic in the medium and you can go for the selection. So that's the story. So in that way you can select the clones, and then finally what you have to do is, following clone selection, you have to identify again the same thing, that is if a gene is expressed or not. Mind that genomic library allows you to identify whether a particular gene is present or not, but expression library, cDNA library, is an expression library. It will allow you to know whether a gene is expressed or not, because all are exonic sequences. So a gene is expressed or not, that is the information which you get in a cDNA library. So you can again go for nucleic acid hybridization. That is, you can go for the preparation of a probe. Any exonic sequences can be used as a probe, and you use that to screen the library, see whether the binding is occurring in that particular, in the DNA which is being represented in one clone. That can be done. Or sometimes people go for immunological or enzymological techniques. Mostly we go for the hybridization procedure because it is comparatively less expensive. So I hope you have got an idea regarding the libraries. So this was the first part of my discussion, that is preparation of DNA libraries. So two libraries, that is genomic library and cDNA library. What are their purposes? Why do we have these two different kinds of libraries? And how do you prepare the libraries? How do you construct the libraries? Okay. The next topic now that I will take up is methods of genetic engineering. Now here there are actually two methods. One is vector mediated method, another is the vectorless method. Now, vector mediated method, that is Agrobacterium tumefaciens mediated method, I am not going in details today, because you please follow my earlier session. Because in one of my earlier sessions, I discussed in detail regarding the procedure for Agrobacterium tumefaciens mediated transformation, what are their advantages and disadvantages and so on. Everything has been discussed. And this is a vector mediated because you have the TI plasmids, which contains the tDNA region, and there you do all kinds of engineering. But sometimes we have to go for an alternative method. Why? Because there may be certain samples, there may be certain plants which are recalcitrant to Agrobacterium tumefaciens mediated transformation. What is meant by recalcitrant? That means where it is very difficult to carry out Agrobacterium tumefaciens transformation, because for a long time it was believed that monocots, rice, wheat, maize, okay, these plants they are very much recalcitrant to Agrobacterium tumefaciens transformation. That means it is not very easy to carry out Agrobacterium transformation. Why? Because of several reasons. Number one is they have certain they have very low content of acetylsyringon, which is a phenolic kind of sensor. Because they secrete acetylsyringon in very low amounts, and also they release certain secondary metabolites which actually hinder the colonization of Agrobacterium tumefaciens to the plant surface. But now people are routinely carrying out Agrobacterium tumefaciens in rice, wheat, etc. It has been well standardized, but it's a bit difficult. So in these plants, you have to go for some alternate strategy or alternate genetic engineering method, and that is possible through vectorless method, that is, which is also called as direct gene delivery method or physical transfer method or physical method of DNA integration. So we will today, basically in this second part, we will discuss about the vectorless method, because vector mediated method I have already discussed. Now, among the vectorless method, the first and the foremost one, which is very interesting, is the particle bombardment method or gene gun method or bialistic method. If you see my slide presentation, you can see I have shown the picture of the instrument, which is a popular instrument from one of the very well-known companies, and this is the bombardment machine or the bialistic machine. So the basic principle of this bialistic process is that it is a helium driven particle gun operating system, and the DNA bearing tungsten or gold particles 1 to 3 micrometer in diameter. That means you have to coat the DNA with certain particles which are usually gold particles or tungsten particles. Normally we use gold particles. They are placed on the leading surface of a macrocarrier. Released from the macrocarrier upon impact with a stopping plate or screen, which catches the macrocarrier. When helium gas is released from the tank, so you can see that this instrument, it has helium cylinder connected to it, and normally this helium gas is actually made to enter into the chamber here, this particular chamber here. So with a high velocity, the helium gas is actually coming. And when helium gas is released from the tank, a disc known as ruptured disc gets ruptured. And the compressed helium gas is released, and the pressure of the gas is tremendously high, 500 to 1700 psi. Of course, you can adjust the pressure. If it is a soft tissue like callus, then obviously the pressure has to be little bit lower. Pressure has to be comparatively lower. If it is a very hard tissue like seed, obviously the pressure has to be enhanced. This accelerates a thin plastic sheet carrying microprojectiles into a metal screen. Upon impact with stopping screen, the microprojectiles pass through the mesh screen. Microprojectiles travel through a partial vacuum until they reach the target tissue. The partial vacuum reduces aerodynamic drag upon the microprojectiles and decreases the force of shock wave created when macrocarrier impacts the stopping screen. So the entire operation is actually occurring within a chamber. So what you are doing basically is you are coating the DNA with gold particles. You are putting it on a particular surface, macrocarriers, and then you are actually releasing the helium pump or the helium gas cylinder, and you are allowing the helium gas to enter into the chamber and under a very high pressure, and under that high pressure you are actually releasing that particular the DNA coated particles which are microprojectiles. You are releasing them, and below that you are placing a Petri plate containing a suitable tissue within which under a high velocity there is an impinging of the DNA coated particles onto the tissue, and so they are being integrated. They enter into the tissue. It's entirely based on mechanical pressure, mechanical velocity. So that is how you are trying to impinge the DNA coated metal particles or gold particles into the sample, and in the process the DNA gets incorporated within the sample. So in a nutshell, this is actually the principle. Now although this method has its own advantages and disadvantages, I am coming to that, but at one point of time when you don't have any option, I mean you cannot go for agrobacterium tumefaciens, which is a more kind of gentle method. You have to go for this kind of mechanical, it's a mechanical method, you can say, depends on mechanical force and mechanical pressure. So obviously it will have its own demerits as well. So some of the points which have to be remembered while you are operating this system is the particles should be of high mass to penetrate into appropriate tissue. The particle size 1 micrometer, tungsten 1.2 micrometer, gold 1 to 1.6 micrometer. Instead, in place of tungsten, gold, you can also use other particles, though they are not that commonly used, like platinum, palladium, rhodium, iridium, second and third row transition metals, and all these metals should be chemically inert because they should not interact with the DNA. So the metal particles that you are using, coating, they should be chemically inert. But mostly you use the gold particles, though it is costly. Second point, certain additives, permidine, calcium chloride, they are used during coating of metal particles with DNA. Target the appropriate cells capable of cell division and competent for transformation. Obviously, you need to choose a proper tissue which you want to bombard. Environmental parameters, temperature, photoperiod, humidity, explants and bombarded tissue all affect the physiology of the tissue. Receptiveness of the target tissue, whether the target tissue is capable of uptaking the DNA. That is also a factor which dictates your success of this technique. Biological parameters, explants derived from plants that are under stress, over or underwater, are all inferior materials. You have to ensure that the tissue that you are taking it from a healthy plant. If you are taking it from a diseased plant or already which is under stress, then your entire transformation will fail. Entire genetic engineering method or the transformation method will fail. So you have to be very careful in choosing the appropriate tissue, healthy tissue capable of uptaking the DNA. So what are the advantages of bombardment technique? It is clean and safe. Introduction of foreign genes into elite germplasm, which is not possible through agrobacterium. You can do it here. Transient gene expression can be monitored and used for engineering recalcitrant agronomic species like wheat, rice, maize. I already told you in case of monocots it may be difficult. Study developmental process and clarify origin of germline in regenerated plants. But the disadvantages: non-homologous integration into the chromosome, multiple copies and some degree of rearrangement. See, you are doing it under high mechanical pressure. You are applying a very high velocity. Obviously, chances are there that there may be tissue damage or there can be rearrangement of genes, some degree of rearrangement, mechanical injury to the tissue, and generation of the chimera. What is meant by chimera? Chimera means which is not expected. Chimera means there can be some cells which have been transformed, some cells which have not taken up the DNA. So in a same, let's say you are using a mass of callus tissue. So some sectors of callus have taken up the DNA, or the bombardment procedure is successful. Some of the sectors of cells they have not taken up. So you call it a chimera, which is also not wanted because the incorporation should be uniform. But if it is not uniform, you call it a chimera, and that is of no use in transformation. So the chances of getting chimera is higher in case of bombardment as compared to Agrobacterium tumefaciens transformation. So if Agrobacterium tumefaciens works, I think it's better to go for vector mediated transfer, which is a more gentle method. But if it fails, then you don't have any option. So that is all about particle bombardment. Tungsten particles are cheaper, but still we don't go for tungsten particles. Gold particles are more uniform in size, show lower toxicity, but they are costlier. But even then we go for gold particles only for coating the DNA. Now some of the other procedures that we use is this will come under number 2, that is macro injection. It is the injection of DNA solution by micropipettes into the developing floral side shoots of plants. Within the floral tillers, there are archisporial cells that give rise to pollen in the developing sac by 2 meiotic cell divisions. Such cells might also be able to take up large molecules such as DNA. So what do you do in macro injection? In macro injection, you just inject the DNA solution. By microtips or micropipettes, you just inject the DNA solution into some of the floral tissues. And then you try to develop the plant. Like as they are saying that in the archisporial cells, which give rise to the pollen, and from the pollen you can actually go for the generation of the plant, transformed plant. But again, macro injection is not that routinely used. For theoretical purpose or academic purpose, of course, we study, but that it's not much put into practice in the laboratory. Electroporation. This is again very routinely used. At least with the bacterial specimens, this is very common method. It's a process where electrical impulses of high field strength are used to reversibly permeabilize cell membrane to facilitate uptake of large molecules including DNA. It uses a relatively high initial field strength with a low capacitance and therefore a short decay time. So actually what you do is you have a chamber called electroporator, and within it you take a sample of protoplast and you apply high or low voltage pulse. The chamber is cylindrical with a distance of 1 centimeter between parallel steel electrodes. The pulse is applied by discharge of a capacitor across the cell. Often you need to use one agent called polyethylene glycol, which can increase the protoplast transformation frequency. PEG is believed to assist the association of the DNA with the membrane. So this is again a very routine method. People use this and it is also very fast. The range of tissues, however, seems to be narrower. You cannot use all tissues for electroporation because there are tissues that are susceptible to DNA uptake by electroporation. So this method is convenient, simple, fast, low cell toxicity and inexpensive to obtain transient and stable transformation in differentiated tissues. However, sometimes it becomes very, very difficult to regenerate a plant from the protoplasts, which have been transformed through electroporation. So there are advantages and disadvantages both. And not all laboratories also have this electroporator. Even particle bombardment is a very costly instrument. It actually costs in crores. So not all laboratories can afford to purchase one such bombardment method. And during our student life, we actually operated the bombardment. We were fortunate enough, I was fortunate enough to handle this instrument. But then again, all laboratories, they are not really, they are that much fortunate to have this instrument. Next is the microinjection. Microinjection is the direct mechanical introduction of DNA under microscopical control into a specific target such as embryo, ovule and meristem or protoplasts, cells or a defined compartment of a single cell. Microinjection is able to penetrate intact cell walls. It is host range independent and does not necessarily require a protoplast regeneration system. So microinjection sometimes this is easier. And you can go for it, where you basically use some glass micropipettes of 0.5 to 10 micrometer diameter tips, and using these glass micropipettes you can inject. You can transfer micromolecules into the cytoplasm or the nucleus. And then you have the liposome mediated transformation. What are liposomes? These are artificial lipid vesicles surrounded by a synthetic membrane of phospholipids. This can be used to fuse with protoplasts using PEG and used for gene transfer. So again, PEG you have used in case of the earlier method. In electroporation also you have used. Sometimes it is used. And sometimes you use it in case of electroporation, and then again you are using it here in case of the liposomes. So DNA enters the protoplasts due to endocytosis of liposomes, and it involves the following steps. So what are the steps of liposome mediated transformation? First is adhesion of liposomes to the protoplast surface, fusion of liposomes at the site of adhesion, and release of plasmids inside the cell. And obviously, as I told you again, that the high efficiency is observed only when you use polyethylene glycol. So polyethylene glycol has a number of utilities in plant biotechnology. If you remember, we also use it for somatic hybridization, that is for protoplast fusion experiments. Okay, you use it in electroporation, and now you are using it in liposome mediated transformation. So what are the advantages of liposome mediated transformation? It leads to protection of nucleic acids from nucleus digestion prior to the introduction into the cellular environment. There is very reduced level of cell toxicity. It is applicable to a wide variety of cell types, high degree of reproducibility, and stability and storage of nucleic acids once encapsulated. But remember that in case of plant system, if you try to analyze which are the major more commonly used methods for plant transformation, then the answer will be number 1, Agrobacterium tumefaciens mediated transformation or Agrobacterium rhizogenes mediated transformation. Number 2, that is biolistics or particle bombardment method. Number 3, electroporation method. So in majority of the laboratories, okay, all over the world and also nationally at India, these levels, usually most of the laboratories, they are actually using these techniques only. That is how to create a transgenic plant. Because for transgenic, you have to follow all these engineering methods. So you have to first learn how to properly select the gene, amplify the gene, and then how to clone the gene, okay, and then how to transform the bacterial cells, then how to transform the agrobacterium cells if you are going for the vector mediated transformation, and then how will you infect the plant cells using the agrobacterium culture, and then you have to select the transformed cells. But if you are going through the direct gene delivery method, then as I told you, the bialistic method, the electroporation method, these are the two most commonly used. Of course, you can also go for other techniques like liposome mediated or the microinjection, but these two are more commonly used, of which electroporation is more commonly used because, as I told you, many laboratories cannot afford the bialistic machine because it is so costly. So learners, so I think I have covered one major area of this recombinant DNA technology. In fact, this unit on recombinant DNA technology which you have in your economic botany and plant biotechnology course, actually I have covered it in 2 or 3 sessions because I considered this unit to be very, very important. And I discussed about the cloning part, I discussed about agrobacterium separately. Today I discussed about the library and the other methods of gene cloning or genetic engineering methods. So I think I have covered 3 hours of sessions on this one single unit. And if you follow each of these teleconference sessions, please go through them repeatedly. Whenever you find, these are all available in YouTube's, and you please go through them, try to match with them with the units. Of course, I have given more details, and then try to understand because this is a very vital topic. And then other topics also in this course I have covered from the plant biotechnology part. That is, I have talked on the different molecular methods which are commonly used in plant biotechnology, including all the blotting procedures and then the hybridization procedures, the RAPD, RFLP. Okay, so all these methods I have discussed in one of the sessions. Applications of plant biotechnology I discussed last year also. I mean, where do you apply the transgenic plants? In case of stress tolerance, in case of, developing superior varieties of plants, nutritionally enriched plants. So what are the different applications of plant biotechnology in plant science? So that has been also dealt. So please go through all these teleconference session, video sessions, go through the units, and if you face any difficulty or you have any queries, please feel free to contact us, write to us, or email us. So we will be there to answer to your queries and solve your problems to the best of our capability. So thank you all. Go through these units, and more or less I have covered the entire of the plant biotechnology modules. I mean this course. Thank you all, and wish to see you again in the next sessions. Thank you.